Hi, my name is Dr. Pardo. I'm a team leader at Imperial College London, and today I'm going to tell you about how the crosstalk between cancer cells and lung fibroblast is shaping the therapeutic response in mesothelioma. Let's first introduce mesothelioma. Mesothelioma is a cancer of the mesothelial tissue, which comes as 4 different flavors: pleural, which is the most frequent, peritoneal, pericardial, and testicular. Pleural mesothelioma is triggered mostly by the inhalation of asbestos fibers that enters into the fabrication of insulation material, and these then go into the lung, to the pleural layers, where they transform mesothelial cells and trigger pleural mesothelioma. 8 to 13% of people that have been heavily exposed to asbestos for a long period of time will develop mesothelioma. But the disease has got a very long latency period, with symptoms appearing 20 to 60 years after initial exposure. While asbestos has been banned in Europe and several other developed countries, it is still being heavily used in the developing world, and therefore we cannot expect mesothelioma to be a problem that will disappear in the near future. In addition, we have now found that other fibers, apart from asbestos, such as carbon nanofibers, can trigger mesothelioma in animal models, and therefore there is the possibility that a new wave of mesothelioma may appear in the population in the future. This disease responds very poorly to therapy, and therefore there is a very poor median survival of between 14.9 months for stage 4 and 22.2 months for stage 1. This very poor survival is not necessarily linked to the growth of the cancer cells, but curiously to the extensive fibrosis that accompanies the development of pleural mesothelioma. And this is because the cancer cells recruit and activate fibroblasts that then go on to deposit a large amount of extracellular matrix, which develop into extended fibrosis tissue. Therefore, it could be seen as a good therapeutic strategy to try to prevent the recruitment and activation of fibroblasts as a way to improve the outcome of patients with this disease. And so this is exactly the strategy that we decided to take. The first thing that we tried to determine is whether patient-derived mesothelioma cells secreted growth factors and cytokines that are known to recruit and activate fibroblasts. And indeed, we found that a number of cytokines known to recruit and activate fibroblasts were modulated in the secretion of mesothelioma cells as compared to normal mesothelial cells. We then looked at the effect of these cytokines on activation and recruitment of naive fibroblasts by using conditioned medium from mesothelioma cells and testing chemoattraction in a Boyden chamber assay and looking at molecular changes that accompany changes in recruitment of fibroblasts by treating cells in petri dish with this conditioned medium. What we can see in the chemoattraction assay is that within 24 hours, mesothelioma cells are capable of chemoattracting fibroblasts, as opposed to normal mesothelial cells or unconditioned medium. And the recruitment of fibroblasts by these various mesothelioma cell lines is accompanied with a change in the phenotype of mesothelioma fibroblasts, which it can be seen by fluorescent microscopy with the shape and the actin cytoskeletal organization of fibroblasts changing upon treatment with conditioned medium. These phenotypic changes are accompanied by a change in the molecular profile of these fibroblasts with increased expression of fibronectin, fibroblast activation protein, and fast smooth muscle actin downstream of treatment with conditioned medium from all mesothelioma cell lines as compared to normal mesothelial cells. The next thing that we wanted to know is what type of activated fibroblast we had. Indeed, there are different flavors of cancer-associated fibroblast, such as inflammatory fibroblast or myofibroblast. So we looked at the secretion patterns of our activated fibroblast using cytokines and growth factor arrays, and what we could see is a change in the pattern of expression of interleukins and growth factors that were characteristic of inflammatory cancer-associated fibroblasts. These fibroblasts are known to exacerbate development of several cancer types, and therefore we wondered whether these inflammatory activated fibroblasts could themselves activate and recruit naive fibroblasts. So we repeated the same experiment as in the slide before, but now instead of using conditioned medium from mesothelioma cells, we used conditioned medium from activated fibroblasts. And what we can see is that our activated fibroblasts are indeed capable of chemo-attracting naive fibroblasts, and they can perform this feat in even more efficient way than mesothelioma cells themselves, with recruitment of fibroblasts being observed from 6 hours onward. This recruitment of fibroblasts is also accompanied by activation of these fibroblasts, with again increased expression of fibronectin, fibroblast activation protein. So therefore what we can see is that mesothelioma cells recruit fibroblasts, activate them, and these activated fibroblasts themselves are capable of recruiting and activated other fibroblasts in a runoff process that is self-feeding. So the question is, what do these fibroblasts change in the phenotype of the disease? And in order to answer this question, we created a mixed culture model, spheroid models with activated fibroblasts here in green and mesothelioma cells here in blue. And what we can observe is that the fibroblasts create a layer around the mesothelial cells. What we expected is that the presence of activated fibroblasts would increase the growth and proliferation of mesothelioma cells. And in fact, we found just the opposite. While spheroids made of mesothelioma cells alone grew exponentially with time, co-culture of mesothelioma cells and activated fibroblasts started with much tighter spheroids, which were inhibited for growth. So if activated fibroblasts do not increase the growth and proliferation of mesothelioma cells, what else can they do? Well, they change the survival of the co-culture. So here, for instance, if we treat spheroids made of mesothelioma cells only with a mixture of cisplatin and pemetrexate, which is the standard of care for mesothelioma patients, we can see that these spheroids respond nicely to the treatment. However, as soon as you treat co-culture of activated fibroblasts and mesothelioma cells, you can see this effect almost totally disappeared, and this is quantified here. This is because the presence of activated fibroblasts decreases the activation of caspase 3 and 7 in all our mesothelioma cell lines. So therefore, there is an anti-apoptotic response that accompanies the presence of the activated fibroblast. So we have activated fibroblasts, but are these fibroblasts capable of a fibrotic response? We address this by culturing our activated fibroblasts until they deposited an extracellular matrix, and following decellularization, analyzing the matrix composition with mass spectrometry. And we then overlaid these changes with a pathway analysis and found that a number of molecules that are known to participate to the fibrotic response, such as several collagens, tenasins, and fibronectins, were increased in the secretion of our activated fibroblast as compared to the secretion of a non-activated fibroblast. Also, what we found, again, is that there was an increased expression of inhibitors of TGF-beta, again pointing towards an inflammatory fibroblast phenotype rather than a myofibroblast phenotype. So what is this matrix doing? Well, this matrix, we could further demonstrate, is in itself capable of activating naive fibroblasts, so that if you plate naive fibroblasts on top of the matrix produced by activated fibroblasts, you end up activating these naive fibroblasts, as you can see here with the increase in fibronectin and fibroblast activated protein when cultured on the matrix produced by activated fibroblasts, as opposed to the matrix secreted by naive fibroblasts. This is also accompanied by increase in the expression of, for instance, PCL-XL, showing that the secretion from the activated fibroblasts have also a pro-survival activity on the activated fibroblast. So I showed you before that the presence of the activated fibroblast in co-culture made the cells resistant to therapy, but did not increase the growth of mesothelioma cells. But now we find that if you add to this co-culture the extracellular matrix produced by the fibroblast, you can now trigger increased growth in the spheroids. Not only you increase the growth of the spheroid, but you now increased even further their resistance to treatment with cisplatin pemetrexate to such a level that the mixture has almost absolutely no effect on cell survival. So we then decided to look at what possible signaling changes were taking place in both fibroblasts and mesothelioma cells in co-culture in order to try to identify potential therapeutic target that would interfere with this crosstalk. And we did this using reverse protein array, looking at change of expression, but also phosphorylation of a number of targets. And what we could find is that a certain number of proteins were regulated similarly in activated fibroblasts and mesothelioma cells in contact with secretion from activated fibroblasts. And these included SARC, which was overexpressed, but also hyperphosphorylation of PGGF receptor alpha, hyperphosphorylation and overexpression of mTOR, and also phosphorylation of sites on AKT that are linked to activation of this kinase. We therefore decided to take a number of drugs that targeted the signaling changes that we had identified, and we decided to test them as single agent, but also at 2x2 combinations in co-cultures of mesothelioma, activated fibroblast in the presence of extracellular matrix, as we found that these combinations were most resistant to the standard of care treatment. And we did this in our several mesothelioma and activated fibroblast co-culture. And here, a white staining means that the treatment is totally inactive, while a deep red means that you get extensive killing. And if we average the response of all these different co-culture, what we can see is that pemetrexate cisplatin combination, again the standard of care for this disease, is pretty inactive. However, what we find is that there are some combinations, such as that of cediranib and vistucertib or saracatinib and vistucertib, which are extremely potent, but also have got very little activity on co-culture of normal mesothelial and naive fibroblasts, which decrease the risk of having systemic toxicity of these combinations. And we decided, therefore, to select these two combinations for further characterization. What we can see is that these two combinations are synergistic with sideranib and saracatinib, showing ZIP scores over 10, showing synergistic interaction at very different concentrations. Moreover, what we could demonstrate is that while the combination of cisplatin pemetrexate was very sensitive to the composition of the spheroid with decreased activity of the drug mixture with increased concentration of fibroblasts, the cedarenib vistusertib and saracatinib vistusertib combination had activity which was fairly constant regardless of increased amount of fibroblasts in the mixture. And therefore we could predict that these combination would have a more stable activity regardless of the stage of the disease, whether early stage or late stage. We then decided to test one of these two combinations in animals, but we didn't know which one would be the most potent. And in order to answer this question, we turned to an AI approach looking at functional networks and how the molecular changes that we identified earlier propagated to the entire signaling network within the cells, and see how our two combination treatment interfered with this propagation. And in order to do that, we collaborated with Kirill Veselkov and used a random walk with restart strategy. We also overlaid onto results of this random walk knowledge about drugs available in the FDA that could target this, with the hope to find even better combinations to test in the future. And in order to assess the correlation between the effect of drugs with the signaling changes, we used gene set enrichment analysis, looking at pathway and biological process changes that are associated with the change of signaling pathway that we had identified. And this enabled us to see that saracatinib and vistusertib impacted much better the biological process associated with the molecular changes that we saw at day 1 and 3 of the interaction of fibroblasts with mesothelioma cells than cedarenib, and that therefore a combination of vistusertib and saracatinib would probably be more potent in targeting these changes than combination involved in cedarenib. And this was again further confirmed by looking at how the combination between the two drugs matched with these changes in phenotype. So therefore we decided to test the combination of vistusertib and saracatinib in a genetically engineered mouse model of mesothelioma in collaboration with Daniel Murphy at the Bitson Institute. And to come to the bad news first, what we found out is that the combination of vistusertib and saracatinib was not very efficient at tackling the disease. But the reason for that was not because the drug combination was inefficient, but because the drug combination was far too toxic to the animals, and we had to put a lot of the animals down due to adverse toxic effect. On the other hand, these experiments enabled us to find out that saracatinib as a single agent was actually extremely potent, and far more potent than cisplatin pemetrexate, the standard of care, in increasing the survival of the mice. While vistosertib also increased the survival of the mice, this was not statistically significant. But therefore, we would expect that a combination of vistosertib and saracatinib would still potentially be synergistic in vivo, but we have to find a different schedule for administration of the drugs so that we avoid falling into the toxicity profile that we've observed. And this is work that we are currently undertaking. So what now? Well, first of all, we would like to answer a question, which is, are the fibroblasts activated by mesothelioma cells the same as the fibroblasts activated by activated fibroblasts? And the reason for that is that when you look at a signature of activated fibroblasts in a publicly deposited dataset for RNA sequencing, what you find is that the phenotype of fibroblast is more of a myofibroblast than an inflammatory fibroblast phenotype. And therefore, what we wonder is whether fibroblasts activated by the first round of activated fibroblasts are myofibroblasts rather than inflammatory fibroblasts. And in order to answer this question, we're going to look at the changes in short terms and long terms of expression profile of fibroblasts activated by mesothelioma cells versus fibroblasts activated by activated fibroblasts. And what we've already found is that the signaling in the cells evolves with time. And this is something that we're now characterizing further and trying to find whether some additional therapeutic targets can be gleaned out of this analysis. Furthermore, the dynamic network propagation approach that we took in collaboration with Kirill Vezikov has already yielded additional FDA-approved drug combination that we could test in our spheroid assays and further in vivo. We will enrich the networks that we base this prediction on with the RNA-seq data that we are now acquiring, and we are also adding directionality to our network so that we have more functional interaction on top of protein-protein interaction understanding. In addition to FDA-approved drugs, we are also testing the potential for food molecules and metabolite combination with approved FDF compound, which is a specialty of Kirill. This is where we are now with this project, and there is left for me to thank the people who have been involved in this research. From my own lab, Dr. Yakinti Chrysokoidou, who did a PhD on this project and who's generated most of the data that I've presented you today. Dr. Matthew Wong, who is now taking the project onward with funding from the June Hancock Mesothelioma Research Fund. And this project would not have been possible without close collaboration with various labs at the NHLI, Imperial College, the CRUK Bitson Institute, the MRC Toxicology Unit, the CRUK Scotland Centre, and the Huang Lab at ICR. If you have any further questions on what I presented today or any other area of research that we perform in my lab, you can either email me or come visit our YouTube channel where I present summaries of our various publications, contact me on LinkedIn, or visit the webpage from my lab at Imperial College London. Thank you very much for your attention, and I'll be happy to answer any of your questions by email. Thank you.